So welcome everybody to our Phenomics webinars in the new structure. Today featuring our live Q&A with Dr. Bipin Pandey over here. Hello Bipin, glad that you join us today.
Yeah.
Very well. Just that you know, this live Q&A belongs to a pre-recorded video that is hosted on our IPPN YouTube channel. You will see the original video to which this Q&A refers at the end of the video or in the playlist that you're currently watching. So if you want to know what we are talking about, feel free to just click the video and listen to it. It's, I think, around 30 minutes of your time, but certainly will pave the ground for this Q&A and the questions herein. A few words on the organizers and myself. Well, possibly didn't introduce myself. I'm Dr. Philipp von Gillhausen. I'm the operations manager of IPPN. And IPPN is also organizer, co-organizer, together with the European infrastructure and phenotyping infrastructure project Emphasis, and its predecessor EPPN 2020, also there as an infrastructure access project, co-organizing this webinar series together with the International Plant Phenotyping Network. Enough of the organizers. Now I want to introduce you to Dr. Bipin Pandey. He is a BBSRC Discovery Fellow from the University of Nottingham and just recently wrote a science paper on and now I have to be very clear, not to say something wrong, on the ethylene signaling in roots in compacted soils. We'll get more on that topic later on, but first I want to take the time to introduce Dr. Pandey here and ask him a few questions so that he can do exactly that. So Bipin, once again, thank you for being here. It's a great honor to have a science paper author as our first Phenomics webinars in the new structure. And can you tell us something about how the story before your science paper was and what kind of career you are currently living and which led you to your discovery?
Yeah, thank you, Phil, for inviting and providing this opportunity to talk about this work. So before we started this soil compaction project, I was heavily involved in the root adaptive responses, particular to low phosphate nutrient condition. So my PhD from National Institute of Plant Genome Research, New Delhi, was entirely based on dissecting the role of root adaptive responses like the root hair elongation, number of root, branching capacity under low phosphate acquisition, and identifying and discovering the key genes which are involved in this process and raising the transgenics and characterizing their roles under low phosphate condition. But fortunately, I received a Newton fellowship during my PhD, and I joined this Nottingham University with Professor Malcolm Bennett, and where we co-discovered this root hair elongation mechanism, which is mainly promoted by the auxin biosynthesis and then the transport and the response machinery. So it provides very mechanistic details of how root hair elongation is promoted under low phosphate condition. And then because I was mainly working on the root hair and the root, and then I thought that because root grows in soil, so why not look directly into the soil, what is going on? Because soil is so much of heterogeneous, you can't control any aspect because the moisture, the nutrient, the air, the microbiota, and everything is so diverse. So this attracted me to work directly into the soil. So the big problem which we started to investigate was the compacted soil condition, and how this compacted soil condition uh, inhibit the root tip elongation and thus blocks the ability of the plant roots to forage for nutrients and water. And from there, this journey started to investigate the signals, the genes which are involved in this process. So yeah, it took nearly 3.5 years to get this thing done.
Which is, I think, still a quite fast pace if you consider how long other researchers might have taken until they reach such a high class publication quality. Bipin, so if I understood correctly, that means that you did your PhD in Nottingham, UK already, and then simply continue to stay there and to stay in the field and to work on signaling in root in, let's say, problematic soils. Yeah, might be phosphorus deficient, might be compacted soil density. Is that correct?
Yes, I did my PhD from New Delhi, India. Just the 3 months time I spent in the University of Nottingham as a PhD student, and then I got a Newton Extension Research Grant for 7 more months. So exactly 10 months I studied as a PhD here, and then I got a Beacon funding for 1 year as a postdoc, and then a Royal Society grant for 1 more year. And now I got this BBSRC Discovery Fellowship. So it was like one by one by one, and that's how I'm working here now as a BBSRC Discovery Fellow.
Very, very seamless transition from one stage to the other. Very good. Are people able to listen to lectures, or are you included in any lectural program in the University of Nottingham, just for people that might be interested to hop by and listen to your work or see your work?
Yeah, actually, I am a BBSRC research fellow. That means I am committed 100% in the research activity. But thankfully, my head of the department had allowed me to start teaching process. So from this semester onward, I would be teaching some work related to the soil environment interactions. So
very good to hear. And yeah, out there for people that are close to Nottingham, that are maybe enlisted as students, you will in the future get the opportunity to listen to Dr. Pandey directly. So take the chance. It's certainly one of a kind. Thank you, Bipin, for getting to know you a little bit better. In the next part of our Q&A, we will be going over some of the questions that have been sent to us via email. For people that watched the video, your pre-recorded talk, and that were having some questions. So first questions coming from Fangbo Jiang. Sorry if I pronounce the name incorrectly, from Chinese Agricultural University. So the first question is, many methods were used in the study, which indirectly proved the compacted soil limited the diffusion of ethylene, resulting in high concentration of ethylene in the rhizosphere, and finally inhibiting root growth. Why can't you just measure the concentration of ethylene in the rhizosphere? I think the concentration of ethylene in the rhizosphere is a more direct indication of the scientific problem this study is trying to prove. And could it better be explained in the natural environment? So measuring ethylene directly in the rhizosphere could be applied more easily in the natural environments, and therefore maybe form a path towards in vivo sampling or recreating the signaling in the field.
Yeah, So yeah, Fangbo, this is really excellent question. So although it's very challenging to directly measure the ethylene in soil pores, which are at nano scale, but yes, we are trying to find a solution for that. So recently we are getting a Felix instrument, which is equipped with the micro needle or syringe. So we can directly impale the syringe into these soil pores, which is either compacted or non-compacted, and we can directly monitor the ethylene level in the rhizosphere. And this setup or equipment is integrated with the CT imaging facility, so we can exactly locate the air pores, which is just beside the root tip, and we can dynamically quantify. So we are getting there, and we are uh, building this process. It's very exciting and interesting to directly measure the ethylene into the soil pores. So
A form of guided sampling mediated through the CT imagery, where you will penetrate into the soil core that is being mounted in the CT chamber, and be able to then see also in the 3D picture where the needle is and in which pore you're penetrating, and then really have a guided image-based method to very accurately sample certain gases in the pores of the soil. Quite interesting. I always find how, and this is just now a follow-up question from myself. I will continue reading out Fangbo's other questions afterwards, but directly related. So I was always, I'm always curious, these soil columns that you put in the CT, how comparable to a natural soil matrix are they? Do you have, often you have to use specific substrates, very artificial soils that somehow do not occur in that form in nature. Sometimes people use soil cores, they extract it directly from nature. So how was it in your study?
That's very interesting question. So in our study, we directly took the soil from the field condition. Obviously, it's a natural soil, but we have to crush the soil to make it very fine, and then sieve it through the sieve it to have like 2 millimeter soil particles, and then you pack the soil in the column. So it can be a 3D printed column where you can easily open the column, and you can uproot the plant and see the root structure. And as well as for CT scanning, you can also use directly a column, which you don't need to open it, because you can obviously image them non-invasively using the CT imaging. So, but we also do this, the coring, where you can directly take the natural soil having no disturbance in the soil structure, and you can image them. So we are now going much closer to the native soil cores, and to see like the highly compacted or highly diverse soil, how the root tip inhibition is going on.
Very good. I just compare that to other methods like MRI, for example, where you sometimes have to use substrates of a certain elemental composition, not to bias your imaging signal or the signal that you're trying to get. And this is not the case for the CT equipment.
No.
And therefore you are able almost to work with natural type soils, at least from the chemical composition, as far as I now got from your answer. Okay, very interesting also for some questions that might follow further down, but I will first continue with a question from China, Agricultural University. So second question was, in this study, you compared the root tip shape to discriminate the effects mediated by mechanical impedance and by ethylene. Is this method widely accepted by other researchers?
So this type of study we have done first time, so going at a finer detail of at the cellular scale in the root tip shape change. So earlier studies, they have taken the whole root cap and not at the cellular level. But I hope that people would like this and people will follow this to have more detail in how the root cap shape is changing, how the central columella cells are changing the shape, whether it is the cell division which is being affected, or it is the cell expansion at the very end of the root tip is being affected by this mechanical impedance. So, so this is a very exciting, and it takes time. It takes nearly 20 to 30 days to get one image of that high quality, because you have to put the root from the soil. You have to clear it till it gets very clear to see under the confocal. So it takes a lot of patience and perseverance to see that of clear structure at the cellular scale.
Very interesting that you mentioned this, that the post-processing time for the CT images is taking really so long.
Yeah.
Did I understand this correct?
No, so this was not for the CT scanning. This is integrated with the confocal microscopy and to
Yes.
see the cellular structure. It would be very costly to see the cellular details using CT imaging.
Yeah, probably costing an image analyst more than 30 days. All right, next question. Compacted soil could restrict the diffusion of ethylene. Yeah, you have shown that both via a model simulation as well as in artificial experiment. That was done, I think, by the University of Utrecht. Is that correct?
Yes.
No, yes, as well. So would the flow of other gases, such as oxygen, also be restricted in compacted soils?
Yeah, that's a very interesting question. We previously thought that it might be the diffusion of the oxygen is also inhibited because the soil pore spaces gets very smaller and restricted. And to confirm this, when we used our hypoxia reporter lines and grew them directly into the compacted soil, we didn't see any change in the fluorescence of the hypoxia reporter. So that confirms that although the oxygen level is getting reduced in the compacted soil, but that is not sufficient to invoke the hypoxia response. So by using several hypoxia reporter lines, we found that we didn't see any change in the hypoxia reporter under compacted soil condition. While if you put that hypoxia reporter in the flooded condition for like 12 hours, you clearly see the massive change in the fluorescence. So that shows that when there is a flooding condition, plant does respond to hypoxia, but in compacted soil condition, it doesn't. So this goes to because if you want to see the hypoxia response, it really requires very less oxygen, sometimes less than 2%. So that might not be the case in the compacted soil condition.
So the gaseous media still allows for enough oxygen to be present at the tip of the root in order not to induce hypoxia reaction in the roots, yeah, which is certainly very important. Yeah, otherwise, I mean, we would have a lot of problems more than here. But of course, I mean, the soil porosity and the network of pores in the soil, which can be quite different in abundance and distribution depending on soil type, whether it's clay, loamy soil, sandy soil certainly has the highest pore volume, which is known, and soil scientists of course investigate also the movement and distribution of different elements through the pore network. And the lesser pores there are, yeah, the more impeded and the lesser connected these pores are, yeah. So the more heavy a soil becomes, the slower is also the pace in which different gases mix or also gases like ethylene and so on can move in the soil. Okay, so a couple of more questions coming from Forschungszentrum Jülich in Germany at the Institute of Bio and Geoscience 2, which is of course heavily involved in phenotyping research, and also the application of different sorts of phenotyping tools. This question comes, so how is ethylene signaling affected by different soil types? And now we are not referring to sand, loam, and clay, but for example, a cambisole, so a really agricultural grown soil, so in different depths. Would you think there might be different interactions with the root in respect of the ethylene signaling, depending on different soil strata with different edaphochemical conditions? Is this being investigated?
Yeah, so this is really a very interesting question, that because the soil is heterogeneous, and so there is a topsoil and then subsoil, which is heavily compacted, and there is a plow pan, which is really sometimes impenetrable for the crop roots. So because of this different soil strata and different mechanical impedance, the diffusion of the ethylene would be changed at a different scale of the soil. So we are trying to investigate this in naturally cored soil in different soil types. So we are arranging those soil cores to see how this diffusion of the ethylene will play a role in root penetration in different setup of soil. So it should hamper, but until we test it, we can't say anything. But it's a really very important question for the natural setup, that how the heterogeneous soil strata would affect the ethylene diffusion and thus blocks the ability of the plant to grow further deep down.
Yes, you mainly addressed now physical properties of the different strata that might affect or will certainly affect the movement of gases there. Could you also imagine that there are chemical interactions that might play a role also in respect to the ethylene signaling there?
Yeah, that's a very interesting question, that as we know that nitrate, which flows down with the water and it leaches down. So the chemical species distribution in the soil would be different. And unlike the phosphate, which is mostly present in the topsoil. And there are other elements where they move through the bulk diffusion. So of course, in this heterogeneous environment, and having a different mechanical impedance, the diffusion would play a critical role, plus the movement of the nutrient will also affect. But we haven't done this exactly how in different nutrient condition, in different mechanical impedance at a different moisture level, the root growth will be affected. But that would be a very comprehensive study to investigate that how it is affecting.
Yeah, definitely challenging, but well, as one finding in research often cascades down to a lot of new research and topics that will be tackled by other researchers. This hopefully does also for this topic. So in reference to this, you mentioned in your talk you were collaborating also with Sascha Mooney from University of Utrecht, and he was somehow investigating the signaling, ethylene signaling response in natural soils. Is that correct? And how could you maybe say a few words on what he was after and what did he find out?
So Professor Sascha Mooney is from our own Nottingham University.
Ah, okay.
He's a soil scientist and soil biophysicist. And he spent almost 20 or 30 years investigating the different soil structure and its property, like biophysical and biochemical property of the soil, its native structure and the microbiota, and how it helps the soil to make its good structure versus bad structure, like in the compacted soil condition. And so he's really uh, nailed down several aspects of the physiochemical and biochemical properties of the soil, and he helped a lot to understand the soil biological chemistry and how the plant root interacts with the soil. So, so yes, we got several inputs from him, and we work very closely because we are in the same department.
Mm-hmm. Very nice. Okay, next question. Did you look at root function in compacted soil? Not only the root length, and I think this you did, but mainly in regards to your previous work with the root hairs and the auxin signaling there. Could you imagine some sort of crossover between what you have done previously on root hair? On root hairs and now on general root growth and penetration in compacted soils? Is there something going on in your mind, forming a nice story between these two elements of your career?
Yeah, that's interesting. So we have a very cool observation that the root hairs, so they clearly elongate more in compacted soil. So in the very beginning, when the seed tries to germinate in the soil, so they need to have uh, the grip to go inside the soil, and that's why they profoundly produce the elongated root hairs. So for the early establishment of the seedlings, and for those mutant which are disabled in producing the root hair, they cannot penetrate even in the topsoil. So there is clear link of the root hair elongation, its encourage capacity for the early establishment of the seedling. However, we also found that in compacted soil, we see more elongated root hairs. It may be because of the higher or heightened ethylene response, which we know control the auxin level, and that's how we see more of the root hair. And plant tries to anchor more in the compacted soil so that it can penetrate harder.
Wow, that's very exciting. That's, yeah. Thank you. And next question would be, how would you imagine the ideal sensor setup to measure in vivo, so in field, maybe the ethylene diffusion in soil matrices, and would maybe stable, or not stable, but would maybe isotopes be a cue leading towards developing methods that enable you really to do the same sampling or the same kind of study you did now in a quite artificial setup in a CT chamber in the field, maybe?
Yeah, so ultimately, we want to translate this research into the field condition, so where we can't control anything. So of course, we have to deal with the natural setup condition, and we can't do the CT imaging, but we can take and bring the natural course along with the plant into the big CT scanner, where we can analyze the soil pore structure, the root growth capacity, the root architecture, plus the ethylene level in the natural condition by using this new instrument. So I think much more can be done, and we can go much closer to the real world by embracing the new technologies and the new knowledge and the new resources. So, and then finally we can translate and this whole idea of having ethylene insensitive, highly compaction resistant crop genotypes in the hands of farmer.
Okay, yes, certainly. I mean, first you discover something in the lab, yeah, then you have to make the transition to the field at some point in order to really prove that what you have discovered also translates to real world, on fully real world conditions, yeah.
Yeah.
And then of course the question will be to further transfer the findings into certain applications. And there you already mentioned that this discovery will open, for example, breeders to now breed for or to use this mechanism of ethylene response to breed for crops that can now penetrate compacted soils, maybe in especially arid areas, yeah, or in areas with soils with a very high clay content, where of course soil compaction is even worsened by environmental conditions or physical properties of the soil, even more than for us here in our comfortable Central European, North European climates. So is there already work being done? Is there already breeders that hop on the train and try to come up with products, seed products for farmers?
Yes. So exactly, some we are in contact with some breeders, and they are very keen. And actually, we are collaborating, and we got some very exciting results in the wheat and in tomato. And in near future, we are planning to test them in the real field condition to have the field trial whether the ethylene sensitive genotypes in crops like wheat, barley can really withstand the compaction challenge. So we are getting more closer, and people are showing huge interest in our work. And so basically, it provides a tool to the breeders to screen the large germplasm very quickly to have this ethylene sensitivity or insensitivity, and then quickly testing those genotypes in the field condition. So this tool would catalyze the breeders and the farmers to have this compaction resistant genotypes more easily than doing the classical breeding.
Just to have some perspective, how big of a problem is soil compaction, and how does it display globally? Is it like I said before, in arid areas or in clay soil type regions? It's certainly more having a bigger impact than here. Can you give us some maybe numbers or indicate how big of a problem this is worldwide?
I'd say it's a global challenge, and it's a increasingly global challenge at it encompassing. So if you say like the uh, Europe, which is a developer, of course, where they use heavy machineries more than the developing world, and around 36 billion hectares of the agricultural land is prone to soil compaction, so which have a severe impact on yield, and it can be around 25%. And due to this intermittent and shift in the climate, if you combine this compaction with the drought, the yield reduction can be as high as 75%.
Wow.
So this is a massive challenge, particularly in the modern agriculture practices, where we have a different tillage practices using heavy machineries for plowing, for cultivating, and for harvesting, which makes the subsoil more compacted, and there is a plow pan like structure which is really impenetrable for the root crops. So so this is a big challenge we are facing currently.
Wow. So it must be massively rewarding to have such a scientific discovery leading to such a big impact. Yeah, I mean, millions of hectares of farmland are being affected by this problem, and well, you paved the way for having a solution to this problem. So congratulations, and yeah, let's go over just to the last few questions. So how will you personally continue your research? Yeah, what will be the next challenge for you specifically? Maybe completely new topics that you would like to tackle in the future. What are your plans there?
So actually, like we are working directly on the plant and soil. So as we know that soil compaction is associated with several other abiotic challenges like the flooding condition. So when there is a flash flood-like condition, the compacted soil doesn't allow the rainwater to go inside the soil because their recharge capacity is blocked. So there is a flash flood-like situation, which is like eroding the soil, which is a potent factor in the soil degradation. So and more likely when there is a drought-like condition, so the water content from the soils get lesser and lesser, and it makes the soil harder. So it mimics like the soil is close to highly compacted, although there is less water. So that we generally say it's that the strength of the soil is increased when there is a drought-like situation. So the question is whether this compaction resistant genotypes can penetrate in the drought more and get more water from the subsoil zones. So these compaction resistant genotype actually can withstand the moderate drought condition, as well as they can help the in the flood situation, as if the roots are growing in the soil. So actually root act as a drill machine. So they will make the a soil more perforated. So even if the compacted soil, the water recharge capacity of the soil would be higher. So the like flash flood like situation can be minimized. So these are the some areas in aspect where I want to put my energy in next. So by just getting first the real compaction resistant genotype and then testing them in a different ecological setups in different abiotic condition.
So directly jumping on the next global challenge that you will try to tackle with this. Funny thing, that would have been my next question, and how far this mechanism can be used to ameliorate soils. Of course, not only for flooding conditions by increasing the macro porosity through the roots that dig into the soil matrices, but for example, in respect also to climate change. Yeah, I mean, there you have you open the way to sequester carbon into deeper soil layers, which is currently considered to be a very good mechanism of carbon capture in natural ecosystems. So this, of course, also plays into that, right?
Indeed, this is one of the most striking aspect to dwell upon by having more sequestration of carbon in the soil and thus reducing the atmospheric carbon level. So can we reduce that by using those deep rooting varieties which are compaction resistant? So it is something which is very lucrative and very exciting at this moment, and we really want to focus in this area.
How would you think are chances, or how would you think that also other plant disciplines might use the discovered mechanism? For example, phytoremediation. Yeah, for example, restoration of grasslands. Yeah, where you can, of course, now that you know that the ethylene response is playing a vital role in how far also natural occurring species, yeah, that happen to grow in the ecosystem, yeah, can penetrate deeper into different soil layers, potentially also making nutrients that have been formerly sealed in these high density or under these high density soil layers can be made available. I mean, this is really, I'm just now playing a little bit with the consequences and impact of this research, also for other plant scientific disciplines. That is really tremendous.
Yeah. So it can be actually utilized to see that what are the labels of different elements, like heavy elements like lead, zinc, and copper in the different soil strata, and whether the plant is able to take them. So we can uh, put those genotypes or the crops or the plants for the phytoremediation uh, tool. So, but it needs to be investigated in the real context.
Absolutely. And what do you think model tools like CRISPR-Cas or gene editing in general? Yeah, let's say it like this, or yeah, what can they possibly do with this with knowing of this discovery? Do you maybe have directly heard from people they will going to use this mechanism and combine it with gene editing or genetic modification methods?
Yeah. So this was one of the question which was most frequently asked to us, that ethylene is such an important signal, and if you are disrupting this ethylene signaling for the whole plant system, so you are actually going to devastate the whole plant growth development, and it can be prone to several biotic challenges if the whole plant is ethylene insensitive. Now we know that which cell type is important. So we are using this cell-specific CRISPR tool to disturb the ethylene signaling in those particular cell types in the root, and the remaining plant is normal for the ethylene sensitivity. So in future, we will really investigate this by creating this cell-specific CRISPR line, which is only affected in like a single cell types in the root.
Yeah, I'm definitely curious, and we'll be looking out for this and what will be achieved via these. So we are nearing the end of our Q&A. However, I still want to give our audience the chance to engage in a direct interaction with you, Bipin. So if there is someone from the audience, please now raise your hand and I will unmute your mic so that you can directly interact with Dr. Pandey. Otherwise, we would be closing up, asking you about maybe also future possibilities where we can see and interact with you and hear more of your research. But first, sure. Is there someone in the audience having a question? Doesn't seem like that. So I will ask you, Bipin, for example, where will we be able to see more of you in the future? Are any conferences booked for you this or next year? Is there maybe even a roadshow happening where you give public talks to larger audiences where people can attend?
Yeah, so I'm planning to have a talk in ICAR conference and in SEB, Society of Experimental Botany. Plus, we are also organizing this Rooting 2021 in parallel with the International Society of the Root Research, University of Missouri. So where I will talk about this soil compaction work and more of the downstream signal which shape the root adaptive responses in the compacted soil. So Yes,
so mark your calendars, everyone. Routing 2021 is this year combined with the ISRR 11. Let me quickly have a look to name the dates. It will be 24th to 28th of May this year, if I'm correct.
Yeah.
So there you will get the chance to hear Bipin live. Fortunately, this event is also co-sponsored by IPPN. So I'm very glad to, well, sponsor here, and I will definitely also take part listening and maybe meeting you there as well, Bipin. Thank you very, very much. Also from the audience, I hear now, thank you for the great discussion. I hope you enjoyed it as much as I did, Bipin.
Yes,
it was
I really enjoyed this, yeah. And looking forward to meet you again in Routing 2021. And many thanks for providing this really good opportunity to talk and discuss about this work.
It was a real pleasure, Bipin, interacting you from the first second to just now. It was, yeah, I wish you every good thing in the world for your future career, for your personal life. And yeah, that I hope that I will meet you again many, many times. It's been really great.
Thank you. Thank you very much.
Thank you also to our audience, and thank you to the organizers of this webinar series, the European Infrastructure Access Call, EPPN 2020, the International Plant Phenotyping Network, IPPN, and the European Infrastructure Project Emphasis. Thank you very much, and see you next webinar.
See you. Yeah, bye-bye.
Bye-bye.